In this session for the advanced cell biology students, we're going to cover vesicle-bound protein transport system, and in particular look at the mechanisms of vesicle fusion and vesicle transport. So you've seen this diagram in the previous lecture. The gated nuclear transport will be covered in a lecture next week. Transmembrane translocators was briefly mentioned in the previous lecture. What we're going to cover today is how vesicles get from A to B within this green box from endoplasmic reticulum to Golgi to the other sites. And we're going to cover various aspects of this over the next 2 weeks. So we're going to start off looking at the rough endoplasmic reticulum, and again this should be stuff that you've covered previously. This is electron microscopy image of the rough endoplasmic reticulum showing all of those ribosomes bound to the rough ER, and those ribosomes are translating protein directly into the endoplasmic reticulum. So this is your classic view of the rough endoplasmic reticulum. In contrast, the smooth endoplasmic reticulum is not associated with ribosomes, and this is an area that's more associated with lipid metabolism and steroid hormone metabolism, so not involved in new protein synthesis. So we are really dealing with the rough endoplasmic reticulum out of these two. So from the endoplasmic reticulum, proteins make their way to the Golgi. So on this diagram there's a rough ER with all the black dots of ribosomes on there, and then from there vesicles are passed to the Golgi apparatus, which is shown in more detail here. And this has two sides. It has a cis face, which is a receiving face of vesicles from the endoplasmic reticulum, so this would be the cis face, and which is here, and then the trans face where there's transport from the Golgi, so that would be this bit here. And vesicles are received on this face, then passed through the Golgi stack, and then transported from the trans face. And as they go through the Golgi stack, they can undergo post-translational modifications, so crucial changes that alter their activity and function, and these are often added as they pass through different parts of the Golgi stack. So from the transface of the Golgi, those vesicles can be released, and where they can then go and fuse with endosomes. Endosomes are just transport vesicles. From here, that material can be passed on to other sites. They could be sent to the exterior for secretion, or those vesicles could be sent to the lysosomes. Lysosomes are the site of intracellular digestion of proteins, so lysosomal enzymes would have to go via endosomes to the lysosomes, and then they will end up in their final destination. We're going to cover more work on lysosomes in the endocytosis lecture, but just for now, lysosomes are low pH, so quite acidic vesicles that have acid hydrolases within them, and they can break down proteins, DNA, lipids, and they are a site where all sorts of proteins can be taken up by the cell and then broken down into constituent parts, amino acids, lipids, cholesterol, that type of thing. Also, you'll find these very, very active in macrophages, and macrophages can take up entire cells and take up bacteria, and those engulfed materials for destruction will be sent to lysosomes for breaking down into nucleotides, amino acids, lipids, cholesterol, and so on. So as vesicles bud from various different places, they have different vesicle coats, and there are 3 different coats that you need to know about. There's a clathrin coated vesicles, and these are predominantly involved in endocytosis, which I will cover next week. They're also involved in sending proteins to late endosomes and lysosomes. So proteins that need to get from the Golgi to the lysosomes will go in the clathrin coated vesicle as well. Whilst we're dealing with the endoplasmic reticulum, the main protein coats you need to know about is COP2. So COP2 coated vesicles bud off from the endoplasmic reticulum and go to the Golgi, whereas COP1 coated vesicles bud off the Golgi and either go for secretion or back to the endoplasmic reticulum, because these vesicles shuttle round and round. So material's constantly going forwards, or the material is going backwards. So COP2 goes from the ER, COP1 goes back to the ER or COT1 for secretion. So those are the 3 coats that you need to know about. So when proteins bud off from a specific site, those vesicles need to know where to go. So if a vesicle buds off from the endoplasmic reticulum, that will be a COP2 coated vesicle, but it will also have other tags on there, and these tags involve Rab proteins which will determine the targeting site of the vesicle. So Rab1 is involved in the ER and Golgi. For example, Rab7 will take a vesicle to the late endosomes. So if you have a vesicle that needs to go to the lysosomes, which goes via endosomes, the proteins in that vesicle will be they will be in a vesicle which has got a Rab7 or Rab8 tag on there. That will take that vesicle to the early endosomes. Rab7 will go to the late endosomes, and then that will fuse with the lysosome, as we'll see in a later lecture. So every single vesicle component of the cell has a Rab tag that will take it there, and these are protein tags on the surface of the vesicles. There are another group of proteins called snare proteins, and these proteins are crucial for vesicle fusion once the Rabs have got the protein to the right place. So the Rabs will get the vesicle to the right place. Once it gets there, the snare proteins will cause vesicle fusion, so that the proteins in the transport vesicle getting to the destination, they will get into that destination vesicle by the 2 vesicles fusing together. And the whole process is shown here. So we've got a Rab protein in its GTP bound form. So GTP, you'll know ATP as a unit of energy currency, well, so is GTP. So a Rab GTP protein is just one that is charged up with GTP, and it is in an active form. So this could be Rab7, this could be going to an endosome, and then on the target membrane there is a Rab effector protein which binds to this particular Rab. So this is basically trying to detect any passing vesicles which have the correct Rab protein on its surface. Once these 2 interact, we've got docking. Then we need to have vesicle fusion, because this alone will not cause vesicle fusion. On the surface of vesicles we have snare proteins, so we have V snares and T snares, and V snares are on the vesicles that have been secreted, and the T snares on the target, and these are very sticky. They're almost like Velcro in the way that they act. So as soon as this vesicle gets somewhere near the target vesicle, then the T snares and the V snares will interact, twist around each other, and force this vesicle in close proximity to this one so that they fuse. Now remember, vesicles are phospholipid bilayers around the outside, and phospholipid bilayers, if you squeeze them together and squeeze out the water in between, they will fuse. And this is shown here. You've got V snares as a single polypeptide chain, and then T snares, which are 2 or 3 chains tightly clustered, and these are very sticky. So when they come together, they will bind, twist, and force this vesicle in close proximity to this one. So whilst they are initially loosely bound, you've got water in between the 2 phospholipid bilayers. As they twist and drive the 2 vesicles together, you drive out the water. And much like if you have blobs of oil floating on water, if you poke them so that the 2 blobs of oil, I'm sure that's not a technical term for oil, but anyway, 2 blobs of oil, if you force them together, they will fuse in exactly the same sort of way. These vesicles will fuse. The phospholipid bilayers will fuse. Initially, the outer phospholipid bilayer of this vesicle and the outer one of this one will fuse together to form what we call a stalk. Then we get hemifusion, and then that's where the inner phospholipid bilayers form a bilayer, and then we get complete fusion, and the 2 vesicles at this point are fused. So hemifusion occurs initially once you've driven out all of that water in between the 2 vesicles, and then you get complete fusion occurring very soon after. Okay, so this is just a summary of the key points that I've covered. Proteins pass through the ER membrane into the endoplasmic reticulum lumen. That was all covered in the last video. From here, proteins are passed to the Golgi in COP2 coated vesicles. From the Golgi, proteins can be passed on either to the cell's exterior, to the lysosomes, and also membrane proteins can be passed to the plasma membrane if they are membrane bound. The vesicles are guided by Rab proteins, so the vesicle has a Rab protein on it that's specific for a destination. These attach to Rab effector proteins at that set destination. Then the snare proteins drive vesicle fusion, and then once the vesicles are fused, then the contents of the transport vesicle fuse with the sort of target vesicle, and then we get depositing of that protein in that destination. Okay, so that's the end of this week's material. In the live session this week we'll be covering the questions that are on at the end of the previous video. Next week we're going to cover endocytosis. We're going to look at how proteins travel through the Golgi stack. We're going to look at specific signal peptides that allow proteins to get out of the endoplasmic reticulum and then sometimes get back in again, and how those proteins interact with the various protein coats. So we'll be covering more detail on clathrin, COP1 and COP2 interactions in next week's videos.